scientists pinpoint immune response behind mRNA vaccine-linked heart inflammation. Researchers at Stanford University have identified a specific two-step immune reaction that explains why mRNA COVID-19 vaccines, while overwhelmingly safe and effective, can sometimes trigger heart inflammation, myocarditis, in a small number of recipients. The findings, published in Science Translational Medicine, shed light on the underlying mechanisms, potentially paving the way for strategies to mitigate this rare but documented side effect. The two-stage immune cascade. The study reveals that vaccination first activates macrophages, a type of immune cell, which then releases the signaling molecule CXCL10. This, in turn, stimulates T cells to produce another inflammatory signal, IFN-gamma. Together, these two cytokines drive inflammation that can damage heart muscle cells and exacerbate further inflammatory responses. This process explains the elevated cardiac troponin levels often observed in affected individuals, a clear marker of heart muscle injury. The risk remains low, roughly 1 in 140,000 after the first dose and 1 in 32,000 after the second. Rates are highest among young men, 1 in 16,750. But critically, the vast majority of cases resolve quickly without lasting damage. Why this matters? Beyond COVID-19, the discovery is significant not only for understanding vaccine-associated myocarditis, but also for broader implications in mRNA technology. mRNA vaccines represent a major leap forward in medicine due to their rapid development capabilities, adaptability to evolving viruses, and potential for targeting diverse pathogens. However, this study confirms that even highly effective medical interventions can have unintended consequences. The researchers emphasize that COVID-19 infection itself carries a far greater risk of myocarditis, 10 times higher, alongside numerous other severe complications. Nevertheless, understanding the exact mechanisms allows for targeted interventions. Key findings. From lab to clinic, the Stanford team used a combination of advanced laboratory techniques, including human cell models and mouse studies, to unravel the immune cascade. CXCL10 and IFN-gamma are central. These two cytokines were consistently elevated in vaccinated individuals who developed myocarditis. Macrophages trigger the response. Macrophages release CXCL10 after exposure to the vaccine, initiating the cascade. T cells amplify inflammation. T cells respond to CXCL10 by producing IFN-gamma, escalating the inflammatory reaction. Blocking the cytokines reduces damage. Inhibiting CXCL10 and IFN-gamma significantly reduced heart inflammation in both animal models and human heart tissue simulations. A potential solution. Genistein. Interestingly, the researchers found that genistein, a compound derived from soybeans, offered protection against heart damage. Pre-treating cells and mice with genistein reduced inflammation, caused by both mRNA vaccination and direct exposure to CXCL10 and IFN-gamma. The study suggests that this dietary compound may counter the inflammatory response, though more research is needed to determine optimal dosage and efficacy. Looking ahead, the findings suggest that heightened cytokine signaling might be a common feature of mRNA vaccines, as the body's immune response to foreign genetic material inherently involves inflammation. While the risk of myocarditis remains low, this study underscores the importance of continuous monitoring and refinement of mRNA vaccine technologies. The researchers also believe this understanding could extend to other mRNA-based therapies, ensuring both efficacy and safety. Your body needs these cytokines to ward off viruses. It's essential to immune response but can become toxic in large amounts, says Dr. Joseph Wu, lead author of the study. This is a crucial balance, and further research will focus on fine-tuning that response.